Path integration. Rather than relying on landmarks, path integration allows animals to return to a starting location, usually their home, by keeping track of the combined distance and direction traveled on a single journey. Path integration, also called dead reckoning, was revealed in a study with foraging desert ants, Cataglyphus bicolor, that leave their nest and take a circuitous route searching for food. As soon as food is found, they turn and run, more or less directly, back to the nest. See figure 5.7. If the nest is moved, ants exhibit a back-and-forth search pattern centered on the previous nest location. If ants are picked up and moved at the beginning of a return trip, they behave as if they were not displaced, running the computed distance and direction that would bring them to the goal, then frantically searching where the goal would have been located. Path integration is studied most commonly in insects, but it has also been observed in birds and a number of mammalian species. This includes mother Mongolian gerbils, Merionones unguicularis, which exhibit path integration during pup retrieval. Although their search involves numerous twists and turns, when the mothers find a pup, they carry it directly back to the nest, even in complete darkness. Figure 5.7. Path integration in desert ants during foraging. The solid line marks the travel path of an ant as it leaves its nest, N, and locates food, F. The dotted line shows the ant's travel route back to the nest. The outbound path is 354.5 meters. The maximal distance from the nest is 113.2 meters. Small filled circles represent 60-second intervals along each path. Okay, so it's basically showing that an ant takes a fairly circuitous path to find its food, but then when it does find its food, it proceeds basically directly back to the nest. Path integration necessitates continuous monitoring of one's present location in relation to a starting point. In other words, an organism must receive ongoing feedback on its own movement through time and space in order to calculate a direct path home. Despite demonstrations of path integration in different species, there is no common formula for how this is accomplished. Even insects use different mechanisms to keep track of distance and direction on each journey. Honeybees use visual flow information, the rate at which images cross the eye surface, to calculate return distance to a food source, whereas desert ants use a pedometer, keeping track of the number of steps they have taken from a starting point. These differences make sense, as bees fly through their environment, whereas ants walk. Spiders use a similar mechanism to ants. At the same time as they are calculating distance traveled, insects monitor the direction of their travel using the sun as a reference point. Amazingly, they are able to compensate for the movement of the sun across the sky, suggesting that some internal clock is marking where the sun should be at a particular time. Details of this cognitive process will be covered in Chapter 6. This is true even when insects are reared under conditions in which they only see the sun for part of the day. Unlike insects, path integration in mammals appears to involve the vestibular system, which is composed of sense organs in the inner ear that monitor body rotation and movement in relation to gravity. For example, rats with vestibular lesions display path integration when the environment is lit, i.e., they travel directly back to a nest after finding food, but appear lost in the dark, searching randomly until they stumble across their home. Unlesioned rats travel directly to their nest in both conditions, although they probably rely on different cues to do so, i.e., visual in the light and vestibular in the dark. Disrupting vestibular signals in other ways, such as slowly rotating an animal's environment, has similar effects, suggesting that this is a primary mechanism of path integration in mammals. A major problem with path integration is that minor errors in calculation can have profound effects on navigation. If an ant begins a homeward journey 10 meters from the nest and is mistakenly oriented a few degrees to the side, they may miss their home completely. The problem is magnified the further the animal is from their final destination. For this reason, many researchers believe that path integration is used during the initial stages of a return trip, but that animals rely on other cues, such as landmarks, to navigate when they are closer to a goal. They may also use landmarks along the way to reset the path integration calculator. This helps to minimize errors that would accumulate along a lengthy route. Environmental geometry. Many ideas about small-scale navigation, particularly how animals locate food in a familiar environment, were altered by a classic experiment with rats, Chang, 1986. The study design, shown in Figure 5.8, involved a rectangular arena in which the corners were marked by distinct olfactory and tactile cues. In the exposure phase, a food reward was placed at a random location in the arena. Rats were allowed to consume part of the reward and then removed from the arena for a short period. When they were returned, the remainder of the reward was hidden in exactly the same place under the sawdust flooring. Rats quickly learned to return to the location and uncover the reward. On test trials, the arena was rotated after the exposure phase, but the cue stayed in the same location. The results were telling. Half of the rats searched in the correct location, but the other half dug through the sawdust in the diagonally opposite corner. This effect, dubbed rotational bias, indicates that these animals were using information about the geometric relationships in their environment, rather than cues such as smell, to locate the food reward. Figure 5.8. Experimental setup and results of a study testing the use of geometric cues in rats. In all panels, the view of the rectangular texting box is shown from above. Filled circles represent the location of hidden food. A. The testing environment consists of distinct geometric cues in that 3 walls are painted black and 1 is painted white. The corners each contain different visual, tactile, and olfactory cues. Numbers indicate the percentage of trials on which rats search in each corner for the hidden food during rotational transfer tests. B. C. All 4 walls are painted black, so geometric cues do not provide unambiguous information about where the food is located. The corners contain distinct cues that are used to locate the hidden food. D. All 4 walls are painted black, but the corner containing the hidden food and the diagonally opposite corner contain no cues. The 2 other corner panels contain feature cues, so the location of the food cannot be ascertained by geometric or feature cues. Numbers in C and D indicate the percentage of trials on which animals search in each corner for the hidden food under these conditions. Subsequent studies tested other animals in similar paradigms, often with mixed results. For example, chicks use feature cues, such as wall color, to locate food in a rectangular room that has been rotated, but if these cues are removed, the birds exhibit a standard rotational bias effect. The same is true of pigeons. They navigate using environmental geometry when features in the environment become more difficult to discern. Eventually, it became clear that a range of species from monkeys to birds to fish exhibit a rotational bias effect under certain experimental conditions. The effects are not limited to rectangular corners. Animals can locate a food reward using different types of angles, the length of major landmarks such as walls, as well as the spatial relationship among objects in the environment. As with beacons and landmarks, some species exhibit a tendency to use one type of geometric cue over another, which probably reflects aspects of their natural environment. For example, pigeons rely on the amplitude of an angle more than the length of a wall to locate food in a parallelogram-shaped room. These may mimic images of cliff crevices where pigeons nest. In humans, the tendency to use geometric versus feature cues changes over development. Toddlers exhibit a rotational bias effect, i.e., they use geometric cues when they are searching for a hidden toy, ignoring non-geometric cues such as the color of a wall. 6-year-old children performing the same task rely more heavily on feature cues. Adults do the same, although they revert to geometric cues if the feature cues are not salient or reliable. Spelk and colleagues argue that only humans show this developmental shift in cue use, although they acknowledge that many animals can use either feature or geometric cues, depending on the experimental setup. The same researchers go on to note that geometric cues allow both humans and animals to establish a new path direction. In other words, the ability to use environmental geometry may be more important for orientation than for navigation. This makes sense in that angles and other geometric cues are view-dependent, so they are only reliable when animals are in the same location. They also change as organisms move through their environment, so they are likely to be used at critical points along a journey, such as, "This is where I should turn." Environmental geometry would also facilitate ongoing navigational processes, such as path integration, because errors along a route could be corrected if animals use this information to orient in a new, correct direction. Despite widespread evidence that animals and humans can use environmental geometry in orientation and navigation, there is considerable debate about what this reflects at the cognitive level. Some argue that many of the experimental findings cited above could be explained by simpler processes, such as associative learning, whereas others propose that the processing of geometric relationships occurs independently of other cognitive functions. No one doubts that meaningful aspects of the natural environment are defined by specific geometric shapes, but how this information is used to guide movement through space is still not clear. Cognitive maps. A classic image in psychology is of a rat running through a maze, like the one shown in Figure 5.9. Behavioral psychologists in the early 20th century, in the early 1900s, proposed that animals reached the goal box in these mazes, successfully avoiding dead ends, by learning to turn down alleyways that led to the food. Tolman, 1948, argued against this interpretation. He hypothesized that animals find their way by forming a cognitive map of the environment as they travel through the maze. According to Tolman, this mental representation included the spatial relationship among landmarks and beacons, as well as the animal's own position with respect to these cues. The concept of cognitive maps fits an evolutionary perspective in that a mental image of the animal's surroundings could facilitate adaptive behaviors such as foraging, nest building, and territorial defense. Figure 5.9, Hampton Court Maze, that was used as a model for spatial learning experiments with rats in the early 20th century, in the early 1900s. Oh wow, okay, so this is quite a complex maze, and then the rat has to get to the center to the food. Okay. One of the strongest arguments for cognitive maps is evidence that animals can plan novel routes to a familiar location. Tolman and Honzik, 1930, demonstrated this phenomenon in the lab using a maze with 3 different routes to the food reward. See Figure 5.10a. During the initial training, all of the alleys were opened, and rats learned to find the most direct route, Alley 1, between the start and goal boxes. Then, a barrier was placed at A. When rats encountered the barrier, they turned around and took Alley 2, the next most direct route. Finally, a barrier was placed at B. This time when rats turned around, they took Alley 3 to the goal box, indicating that they understood Barrier B was blocking both of the first 2 alleys. Tolman concluded that rats had formed a spatial map of how the 3 alleys fit together. Behavioral psychologists, such as Hull, explained the findings in terms of stimulus-response learning, pointing out that rats could solve the maze using cues in the room, e.g. lights or pieces of furniture, as landmarks or beacons. Dozens of papers were published over the next 50 years, purportedly supporting or contradicting the existence of cognitive maps. Figure 5.10b shows an updated version of one of these studies using hamsters, Chappius and Scardigli, 1993. Animals were trained to take one route, e.g. A, F, E, but then barred from this route during testing. Importantly, the ability to use landmarks was minimized by rotating the maze and starting the animals from different locations on each trial. Even with these additional controls, hamsters were more likely to use the most direct route, e.g. A, G, E, during testing, suggesting that they had formed a mental representation of the maze design. Figure 5.10, Design of apparatus to test cognitive maps in lab animals. A. Rats are allowed to freely explore a maze that has different routes from a start box to a goal food box. Over training, they select the most direct route, 1, between the two. When a block is placed at A, they select the next most direct route to the food, route 2. When a block is placed at B, rats select route 3, suggesting that they have a cognitive map of the entire maze, understanding that block B obstructs route 1 as well as route 2. Tolman and Hosnick, 1930. B. Hamsters are trained to navigate a circular maze from a start box A to a goal box, e.g. B, where food is located. During training, the arms leading to the center of the maze are blocked, so animals must traverse the circumference of the maze to reach the goal box. During testing, the blocks to the center arms are removed. Animals are placed in A with the path around the outer maze blocked, so that they must travel to the center of the maze. From the center point, hamsters then take the most direct route to the goal box. Chapius and Scardigli, 1993. Other findings support the idea of cognitive maps in different species. For example, if chimpanzees, pan troglodytes, watch an experimenter hiding food at various locations in their enclosure, they retrieve the food in a different order from which it was hidden. That is, they appear to have a spatial map of each location and take the most direct route from one hiding site to the next. Vervet monkeys do the same thing, although they cannot remember as many locations as the chimps. The water maze task, successfully navigated by rats, mice, guinea pigs, and other animals, provides indirect evidence for cognitive maps in that animals go directly to the hidden platform from new star locations. Certain behaviors in the natural environment can also be explained by cognitive maps. Meerkats, Suricata suricata, escape directly to the nearest hole when they hear an alarm call, and honeybees take novel shortcuts between hives and feeding stations, suggesting that both species know the spatial layout of their environment. Reminiscent of the chimps in Menzel's study, marsh tits, Poecile palustris, do not follow the same path when they store and retrieve seeds. Shuttleworth and Krebs, 1982, indicating that they know where the seeds are stored, not just how they got to the storage sites in the past. Finally, jumping spiders, Portia labichia, select the most efficient route to a food source when obstacles, such as broken branches, block the shortest path. The concept of cognitive maps is appealing because many people conjure up a mental image of their spatial environment when they give or receive directions. If animals could do the same thing, it would provide further evidence for cognitive maps in non-human species. The process of giving and receiving directions has been studied extensively in honeybees who communicate the location of a food source through an elaborate dance. Chapter 12 discusses honeybee communication in detail. Gold and Gold, 1988, capitalized on this knowledge by placing a sugar source on the shore of a lake within the foraging area of a nearby hive. Bees who flew to the feeding site transmitted information about the sugar location to the rest of the hive. The sugar source was gradually moved to the center of the lake, and some of the bees transported with it. When foraging bees returned to the hive, again communicating the location of the food source, nest mates did not fly to the new site. According to the authors, honeybees knew that a sugar source was unlikely to be situated in the middle of a lake because they possessed a spatial map of their environment. The study was often cited as evidence for sophisticated cognitive abilities in insects, but the findings were never replicated, a key component of scientific research. In fact, the lake study, as it became known, was never published itself, just reported in other review articles by the authors. Most importantly, when the experiment was repeated, honeybees followed directions to a sugar source in the middle of a lake just as frequently as to a land location the same distance away. At least in this study, honeybees were not discriminating between food located in probable and improbable locations. It is unlikely, therefore, that these insects were navigating using a cognitive map. The insect studies aside, many people are willing to believe that mammals or birds possess cognitive maps, but the evidence supporting this claim is not irrefutable. As with Tolman's initial experiments, Critics point out that rats and other animals can navigate mazes using some combination of environmental cues or a sophisticated form of path integration, even if they have not formed a mental map of their environment. Similarly, birds and primates that retrieve food in a different order from which it was hidden may simply go to the closest cue, retrieve the food, and then proceed to the next cue. Even if researchers agree that a particular species can form a cognitive map, they may disagree on the nature of this representation. A cognitive map may be a precise record of how spatial elements, e.g., cues and geometry, are connected in the environment, a more general association between landmarks and the organism's position within this space, or something in between.